So hello and welcome to another class of Behaving Bio Sciences. This is episode with you. So today I'll be talking about restriction enzymes, which are very essential for your recombinant technology or molecular biology experiments or any kind of cloning experiment. So we heard about most of the things about type 2 restriction enzymes, but I'll tell you the other three, means total 4 restriction enzymes which are present and what are the functions of them, and brief discussion about their detailed function. So let's start. So first one, the four types are type 1, type 2, type 3 and type 4. Sorry, type 2s. Type 2s. So these are the four restriction enzymes which are present and which have a cleavage restriction modification activity on the genome. So first start with the type 1 restriction enzyme. So type 1. So type 1 restriction enzyme consists of one enzyme and three subunits. That one enzyme consists of three subunits with different functions. Suppose A, B, C. So one is for cleavage, one is for modification, another one is for the specificity. So these three subunits consist a single enzyme here, and in real cases, HSD R for restriction, HSDN for modification, and HSDS present for specificity. So HSD RNA system is the one which is regulating this type 1 type of restriction enzymes. So they recognize a single sequence as a modifiers or restrict them, but it cleaves around 1000 dp away from the recognizing or the specific site. So that's why we are not using this in our molecular experiment. But type 1 restriction enzyme is also very, very essential. I will give you an example like the DAM methylase is a kind of type 1 restriction enzyme which controls by HSD RNA system. So, for example, DAM methylase, whose function is the for restriction, not only restriction but modification. Mainly it is known for the modification or methylation of the genomic DNA, but it falls under the type 1 restriction enzymes. The type 2 restriction enzymes are the most important restriction enzymes which are essential for your molecular experiment or cloning experiment or recombinant DNA technology related to any kind of work. So first I will tell you type 2 restriction enzymes is a two enzyme complex. Actually, it is not a complex, it is a two enzyme which you can say type 2 enzymes. Why two types of enzymes? First one is responsible for restriction only, another one is for modification. So that's why two types of nomenclature is also there. Likewise, I'll tell you. Suppose SMA1, that is isolated from Serratia muscicans, SMA1. If it is restriction, the work of that enzyme is only to restrict, that is R SMA1. And if it is for modification, then you can call it M dot SMA1. So type 2 enzymes are two different enzymes, restriction and modification. Now we know they are very specific to their cleavage site and they recognize symmetric sequence or you can say palindromes. I'll give you an example of a palindrome. GATTC is a recognition site for EcoR1. That's very common. You know that already, I think. GAATTC. So you can see that from the reverse direction, the same sequence is here. So this kind of sequence are called palindromes. This palindrome sequence are recognized by the type restriction enzymes, either for modification or for restriction. So it's a very important enzyme for your molecularity work, and widely distributed in different sections. It can cut a particular sequence as a cohesive end, which will generate a cohesive end, or a blunt end. So I'll show you here only in this sequence. Suppose one restriction enzymes, like EcoR1, you cut in this A sequence, is the recognition sequence. So it will cut here and here. So it will generate a 3' overhang and a 5' overhang in this case of this kind of cohesive end restriction of the restriction enzyme. So EcoR1 is the enzyme which generates cohesive ends because of this recognition site or recognition sequence, which will in turn generating the particular sequence. Suppose a separate restriction enzyme, suppose not EcoR1, separate restriction enzyme, but have the similar recognition sequence, but it made a cut in the middle position in this A. So here. So this kind of restriction we will call a blunt end restriction, and this kind of restriction is called cohesive end. So it depends on the type of restriction enzyme and the number of restriction enzyme format is isolated. So it depends on the characteristic of the particular restriction enzyme, and this is under the type 2 restriction enzyme. So type 3 restriction enzyme is also useful information or useful for cellular work. So it consists of one enzyme and two subunits. Okay, so what are the functions? Suppose this is the two subunit of an enzyme complex. So this is the enzyme complex and two subunits are there. So one is responsible for specificity and modification, and another one is responsible for cleavage or restriction. So these two subunits, but present in one enzyme complex, and the function is likewise distributed in two subunits. That is totally different from the type 2 enzymes, and little bit similar with the type 1 enzyme. So type 1 enzymes has three subunits, but it is a two subunits. So it is recognizing, or it will recognize a single sequence. It recognizes and methylates the same sequence, but restriction will be 24 to 26 pp away. So the recognition and the methylation sequence will be the same, but restriction will be around 24 to 26 pp away from the recognition site. This is the disadvantage for your recombinant DNA technology work. But it has several other mechanisms, dependent mechanisms, which are important for our normal cellular function or bacterial cellular function. So I'll give you an example like ECO P15 is the example of type 3 restriction enzyme, which has an important part to play inside the ECO, means ECO Escherichia coli. So type 2S, so it has two different enzymes, same as type 2, two different enzymes. Restriction and modification purpose, and it recognizes an asymmetric sequence asymmetric sequence. The recognition site, it will cleave one side of the recognition sequence. It will cleave the one side of the recognition sequence, and that will be around 20 BP away. So 20 BP away from the recognition site, this enzyme has the specificity to cleave. So it can be any sequence, but it is 20 BP away, around 20 BP away from the actual recognition. So that's why you do not use this enzyme as well in molecularology or molecular cloning or any kind of recombinant technology work. But this is also have a specific function in different bacterial cells and other cells. So I'll give you an example. Fork 1. It's a kind of type 2S restriction enzyme. Here S is for the specificity or binding ability. Specificity. So S denotes the specificity, type 2S, but it has several differences with the type 2 enzyme. So these are 4 kind of restriction enzymes which will be helpful for your study. I think if you have any questions, please ask me in the comment section. Thank you. So if you like my classes, give a big thumbs up and don't forget to subscribe my channel because these things stimulate me to take futuristic classes for you guys. So thank you and be with me.